Okay, welcome back to biology, to general biology. We are now in chapter 5, and this is the last chapter we will cover before your first exam. Your first exam will cover chapters 1, 2, 3, 4, and 5. In chapter 5, we are talking about the structure and function of large biological molecules. There are 4 that we're going to talk about: carbohydrates, lipids, proteins, and nucleic acid. And these 4 biological molecules are the big things that do the things that make our cells alive. These are really big molecules, so sometimes they are called macromolecules. Macro means big, so they can be very large and complex. For example, a protein can have a molecular mass or a molecular weight of over 100,000 Daltons easily. Large biological molecules have unique properties. Those properties are going to range from the orderly arrangement of their atoms. So while all these different biological molecules are based on carbon and hydrogen, they're arranged in different ways that they're going to have different shapes and different functions. When we're talking about macromolecules are polymers that are built from individual monomers. There's an Amoeba Sisters video linked to this chapter that I think does a really good job of explaining polymers and monomers. So monomers are individual little building blocks that are used to build things. And when individual monomers are put together, you have a polymer. Polymers are made by combining monomers or short polymers together through a dehydration reaction. So dehydration means it pulls water out. If you're dehydrating something, you are removing water. So you're going to dehydrate these two things, take away their water molecule, and these bonds are then going to, instead of being bonded to hydrogen and hydroxide, these 2 electrons are going to bond with each other, and you have a larger polymer. This is true no matter what biomolecule we're talking about. Dehydration will link them together, and hydrolysis will break them apart. So if you need to break down a molecule, your body will take a water molecule. So hydrolysis, hydro means water, lysis means breaking. Hydrolysis is taking this molecule and breaking it using water. So you've broken this bond and put in a hydrogen and a hydroxide. This is how you break the bonds of all your biomolecules. 3 of the organic molecule classes are based on polymers. Carbohydrates, proteins, and nucleic acids all have a monomer. Our other category, lipids, it's debatable whether we want to call them monomers or not. Your textbook does not. Enzymes are specialized macromolecules that will make these reactions happen. So water isn't all by itself going to pull itself out or insert itself. You're going to have enzymes that are going to make this possible. Enzymes are specialized macromolecules that speed up chemical reactions, whether we are talking about dehydration or hydrolysis. There are enzymes that do both. A dehydration reaction is when you take 2 monomers and put them together by losing a water. Hydrolysis is when you take 1 monomer and break it apart. Basically, these are backwards from each other. Okay, let's watch this video.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom.
So this purple molecule that's making this possible is our enzyme, that enzyme we just talked about.
To form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
So there, that was another enzyme that was taking those apart. When it says it's catalyzed by, that means it's creating that reaction. Okay, so we're going to go through these 4 types of biomolecules. First, let's talk about carbohydrates. Carbohydrates are really fuel and building materials. That is the 2 basic jobs that carbohydrates serve in our bodies. Carbohydrates can exist in 3 classes. One is monosaccharides. Monosaccharide is going to be our monomer here. 2 monosaccharides combine to make a disaccharide, and multiple monosaccharides combine to make a polysaccharide. Here is some examples of different monomers, different monosaccharides. Trioses are 3 carbon sugars. Pentoses are 5 carbon sugars. Hexoses are 6 carbon sugars. You've probably heard of a lot of these. So our trioses, we have glyceraldehyde and dihydroxyacetone. We are probably not going to talk about these much through the semester. 5 carbon sugars, we have ribose and ribulose. Ribose, of course, you've heard of in RNA, ribonucleic acid, or deoxyribonucleic acid, DNA. Ribose is found in those. Hexoses, we have glucose, galactose, fructose. They are all different isomers of C6H12O6, so they're just in different shapes, whether they're aldehyde sugars or ketone sugars. And these you've probably heard of, you know, when you just eat food, as you've eaten food all your life, you've probably heard of these sugars through there. Regular, like fructose is fruit sugar. Monosaccharides all have this molecular formula, CH2O. So if you have 3 carbons, you have 6 hydrogens and 3 oxygens. If you have 5 carbons, you have 10 hydrogens and 5 oxygens, CH2O. If you have 6 carbons, you have 6 hydrogens and, if you have 6 carbons, you have 12 hydrogens and 6 carbons. Monosaccharides are the simplest of all sugars. Monosaccharides will be found between 3 to 7 carbons, although these are the most common sugars we're going to talk about. And sugars are classified by the position of the carbonyl group. So if they are aldoses or aldehyde sugars, which have their carbonyl on the end, or if they are ketoses or ketone sugars, which have their carbonyl in the center of the carbon chain. They're also classified by the number of carbons. So trioses, tri means 3, a 3 carbon sugar. Pentose, pent means 5, so these are 5 carbon sugars. And hexoses, hex meaning 6, these are 6 carbon sugars. So, here we go. Triose, 3 carbons. Tetrose, 4 carbons. Pentose, sugars with 5 carbons. Hexose, sugars with 6 carbons. And heptose, sugars with 7 carbons. You're already familiar with some of these prefixes based on when we talked about branching and size of carbon chains. And now you can see if you turn it into ose, this indicates that you have that C double bonded to O, you are dealing with sugars. So sugars are going to end with OSE, whatever sugar we're dealing with. Glucose is the most common monosaccharide. It is C6H12O6. Molecularly, you could draw it out like this, but in a cell, it's going to pretty quickly form a ring shape. This oxygen and this carbon are going to react, and you end up with something like this. Again, instead of drawing out all those carbons, you just assume that each of these carbons has, each of these angles has a carbon in it. The main purpose of monosaccharides is going to be fuel for cells. So your cell can break this down and get a lot of energy and create ATP. Sugars are also used as raw material for building molecules, and they can also be combined to make complex carbohydrates. When glucose forms a ring structure, this oxygen is going to be either trapped above the ring or below the ring. And this is going to make some difference in how those monomers are going to attach. And it's also going to make some difference in how our bodies are able to interact with those polysaccharides, and even determines what sugars we're able to digest or not. So here in this sugar, glucose, this hydrogen is pointing down. So that makes it an alpha sugar. It's an alpha glucose. Here, the hydroxyl is pointing up. That makes it a beta sugar. So that's when you see alpha and beta in our sugar conversation, it refers to the shape of that hydroxide. The hydroxide is pointing down for an alpha or pointing up for a beta. Glucose, galactose, which is part of lactose found in milk, and fructose, which is fruit sugar, all have the same different chemical formula, but they're going to differ in the arrangement of their atoms. So fructose is a structural isomer of glucose and galactose, and glucose and galactose are enantiomers of each other. You can see just this is right here is where they are different. Then if we join two monosaccharides together, we get a disaccharide, and these join through a dehydration reaction. When two sugars are bonded to each other, this is called a glycosidic linkage. And we can number the sugars. If you start with an oxygen and go around the circle, you can number the sugars. This isn't something you have to be worried about being able to do at this point, but that's why this is called a 1,4 glycosidic linkage, because the oxygen is combined to the number 1 carbon here and the number 4 carbon here. So it'd be 1, 2, 3, 4, number 4 carbon. Here, if you started the oxygen, this is the number 1 carbon, and this is the number 1 carbon, this is the number 2 carbon. So this is a 1,2 glycosidic linkage. And so you can see glucose and glucose combine to make maltose, glucose and fructose combine to make sucrose. So you use a dehydration reaction to create your disaccharides. So disaccharides include maltose, which is part of beer, malt sugar, sucrose, which is table sugar, and lactose, which is milk sugar. Lactose is made by combining glucose with lactose. We don't have galactose on here. Okay, so lactose is made by combining glucose and galactose. So let's watch this video about sugars.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Okay, and then let's talk about polysaccharides. So where our monosaccharides and disaccharides are mainly fuel, your cells are going to burn them for energy. Polysaccharides are mainly going to be either storage, so you store sugar for long term, so that if you are eventually in a famine, you have some stored sugar for energy, and also structural roles, so holding together the cell walls of our cells. The architecture and function of a polysaccharide are going to be determined by what type of sugar monomers it has, and also by what type of glycosidic linkage it has. Here's 3 different polysaccharides we're going to talk about. Starch. Starch is a way that plants store energy. So potatoes are a plant, they have a lot of starch in them. Glycogen is the way that animal cells store energy. So in your body, you store about a day's worth of energy in glycogen. And cellulose is a type of sugar that is found in a plant cell wall. So plant cell walls are made of cellulose. This is a structural type of sugar, and these are storage types of sugars. They're all polysaccharides, and the difference in them is they're made of different sugars, and their sugars are combined differently. So starch is composed entirely of glucose monomers, and these are typically stored within chloroplasts and other plastids in a plant cell. And the simplest form of starch is going to be a compound called amylose. Let's watch this video about polysaccharides.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Okay. Glycogen is a polysaccharide, the storage polysaccharide in animals. We normally store glycogen in liver and muscle cells, and then we can take this glycogen and just break off individual glucose monomers off of it. So you just break off little sugars when you need sugar in your body. So if your blood sugar is low, you haven't eaten in a while, you need some energy, you just break off little sugar from glycogen, and you suddenly are able to transport that sugar around your body. Cellulose is the major component of plant cells. It's similar to starch. They're both glucose linked together, but they're linked together differently. Here you can see all of these linkages have the oxygen facing down. Here you can see that the linkages are facing up and down, up and down. So starch is all alpha glucose, and cellulose is a 1,4-beta glucose. That difference all by itself makes you able to digest starch and not able to digest cellulose. So your body is going to digest this starch into individual glucose and eat it as sugar, whereas this cellulose is what we usually refer to as fiber or roughage. It passes through your body undigested. It's very important for intestinal health, and some of the microbes in your intestines may be able to digest some of it, but human cells can't digest this cellulose at all. Okay, so this is a little bit more talking about what we just said. Starch is, because of those linkages, starch is going to be largely helical. You can see it's rotating around like this. Cellulose molecules are going to be straight and unbranched, which is pretty important for it to be able to become a structural molecule. It can form these important, long, strong threads that end up forming a cell wall. Whereas this just needs to, wind up and be stored in it tightly in a chloroplast. Okay, so the enzymes that digest starch by hydrolyzing the alpha linkages can't digest the beta linkages in cellulose. So this will pass through your body as insoluble fiber. Some microbes will be able to digest cellulose. And so many herbivores, whether we're talking about cows or termites, they're going to have a symbiotic relationship with gut microbes that are able to digest these. And even, a cow will even pass it through their, they have a little bit more complexity in the way that they pass food through their digestive system. Chitin is another storage polysaccharide, or I'm sorry, I said that backwards. Chitin is another structural polysaccharide. Chitin is a polysaccharide that is found in the exoskeletons of arthropods. Arthropods would be insects. It would be things like lobsters and crabs. It would also be arachnids like spiders and ticks. Chitin is very strong and flexible. So here they have made a surgical thread out of chitin because of how strong it is. Chitin also is structural in fungal cells. So the cell walls of a fungus, whether we're talking about yeast or mushrooms or molds, these are made of chitin. Okay, I'm going to pick up with lipids in part 2. So if you miss this lecture in class, or if this is what you don't understand, make sure you come back to chapter 5.2 lecture. I'll see you in a little bit. Bye.